Hello class! Today we're going to be talking about DNA replication, and this is just sort of a pre-lesson video where I'm going to be explaining exactly what goes on when DNA replicates. And during class what will happen is we're going to do biology theater and we're going to act out this process. And in the description of this video what you'll see is a link to a Sign Up Genius, and that will allow you to sign up for any of the enzymes or different parts of this process so you can be a part of replicating DNA in our very own classroom. So let's get started. We have DNA replication, and it starts with the origin of the replication site, and we get an enzyme here called helicase that binds. First you have to have topoisomerase unwind it, but helicase will start to unzip the DNA here, and you get single-stranded binding proteins attaching to that DNA and keeping them apart from each other. And so you have DNA polymerase, and it of course needs to only add to one strand. And so that's the point of helicase unwinding it, is DNA polymerase can only add to one strand at a time. And in addition, for DNA polymerase to start binding, you have to get primase. And it's going to add a little RNA primer, and that's going to sort of signal DNA polymerase starting to be able to add the nucleotides, of course, in the complementary fashion. You have your adenines binding to thymines and your cytosines binding to guanines. Remember, we are making DNA, not RNA, so you do not have uracil in this equation. On one side of the DNA strand, you have your leading strand, and that gets synthesized rather quickly, as you can see, in a pretty linear fashion. In addition, you have another primer that sort of takes away these little RNA bits and replaces them with DNA, but we'll come back to that in a second. For now, I'd like to talk a little bit about the lagging strand, or the other side of the DNA. And on that side of the DNA, you have to make fragments because, again, this DNA polymerase can only add in the opposing direction, the 5' to 3' direction. But this strand is going from 5' to 3' this way, and it can't add 3' to 5'. So it does add 5' to 3', but it only does it in little bits. You get continual addition of these primers, and you get what are called Okazaki fragments, which have to be bound together by DNA ligase. And this continues happening. You get single-stranded binding proteins continually adding and then leaving as more DNA is created. And as it's created in the double strand, it will start to return to the double helix. In addition to this, you have another DNA polymerase 2, which checks for errors in the DNA, which is really important because if you get a nucleotide in the wrong place, or you get some sort of copying error, you're not going to be able to make the proteins that your body needs to function. And so this is just quick DNA replication. If you'd like to go over this process again, I know it's pretty quick and it's pretty complicated with all the different proteins we have and enzymes that are working together to make not one but 2 copies of our DNA from one strand. So feel free to go over this video and be sure to sign up for your part in our play. Thanks!